have you. Okay, so I guess you count yourself among the survivors. The fact that you're still here, even though I haven't posted the grades. That's not to say I know anything about them. I'm working on them. I've got them. I should have the grades posted. I would expect sometime tomorrow. If I read the calendar correctly, you have till 5 p.m. on Friday to decide whether you're going to finish this marathon or opt out. Otherwise, we're just down to one more exam.
It's Friday of next week.
Is it?
Yeah. It's what it says.
Oh, did they? Whatever's on the FAU calendar. Do you have another? So you have an extra? It's not this Friday?
That's in your syllabus.
Whatever the academic calendar says, because they're going to control it. So you're going to have your 3 grades. So here's what you want to think about. You're going to have your 3 grades. If you figured out how to prepare for these exams and you've got 2 good grades and 1 low grade, then you're in pretty good shape. You can think about the final exam. If you got 2 very low grades, most likely you have 3 low grades. I'm not trying to be mean. It's just the math. Then it becomes a math problem. I don't know. I may have said this. I don't know. It's just so many years go by. But even had a student come into my office and asked if I was very good at figuring out what she needed on the 4th exam to be able to get a C in the course. It's like, yeah, I've done it a few times. Okay, so we just started the exam 4 material at the end of the last lecture, which would have been last Wednesday. This is chapter 17 on gene regulation in eukaryotic cells. I'll go very quickly through the first few slides to just kind of reorient us back where we were. The more detailed discussion is available on the video that's up on Blackboard. So what we're going to have is chapter 17, which is on eukaryotic gene regulation. And that's really where we're putting our emphasis, because that's where most of the research is going on. For those of you interested in health career fields and such, and what the impact of genetics and personalized medicine, it's all going to be based on how eukaryotic genes regulate gene expression. It's different. We talked about that. You don't have operon type situations. So we'll talk a little bit about how genes that need to be expressed coordinately, that is expressed at the same time, even though they're scattered around the genome, can get turned on and turned off at the same time in eukaryotic cells. You see a very fundamental similarity. It all comes back to protein DNA interactions and protein-protein interactions. And some of what we're going to be seeing on this will take us back to some earlier chapters as we tie back into some of the material when we talked about gene organization and such. Now we're going to go back and ask, we had that whole chapter, and that at the end we had that basal transcription apparatus and such. We'll come back and revisit that, but talk about now how it regulates gene expression. Then we have chapter 18, which we should start today, which is on DNA mutations and DNA repair. This is probably an area that you haven't seen that much exposure to, except for the occasional base change that changes a nucleotide and changes amino acid. So it's a long chapter. It's pretty complex in some areas, but I'll give you a little more information once we get to that. So that's kind of the big overview of what we're trying to do for today. Before we get started on the actual lecture material, let's do our questions that we've been doing. And while I have those up here and while you're reading through that, so keep in mind when I post the grades probably tomorrow, that average does not include these iClicker questions. So those of you who are coming here, and especially if you're putting in responses to every single question, so you're getting the participation points, most likely will be able to add probably at least in the range of 5 to 7 points onto whatever that average is that I post tomorrow. So, an 84 becomes a 91 kind of thing, okay? All right. Is there a question? All right. So does this question look at all familiar? No? It's just a rewrite of a question we did last time. All right. Oh, I'll go to 1 minute and 45 seconds on this one. Okay. So, do you have to know anything about tyrosine hydroxylase that I haven't told you up here? No? I've just simply told you it's a gene that's expressed in the dopaminergic neurons which produce dopamine. Last time I just said we were talking about insulin gene and we were talking about expression, except I said in brain tissue. This time, now, did we expect to see the insulin gene expressed in the brain tissue? No. Do we expect to see the TH gene expressed in the dopaminergic neurons? Yes, it tells you everything you needed there. So what's the answer? All right. So I know everybody went back and reread everything right after the exam. So the idea is, and we're going to go through the slides real quick, is you see two major events happening when the genes are getting set up to be expressed. You have this relaxation of the chromatin, which makes it more sensitive to DNase if you do these DNA sensitivity experiments. So there would be a hypersensitive region there to the DNase, and you also find low levels of methylation. Okay? But it's essentially the same. It is the same question. I just changed the players. I don't know. Okay? So this one's from today. Although you might know it because we've probably mentioned it a number of times before. This one probably just needs 1 minute. All right. So where do you find the GAL4 protein? Yeah. So it's a rather novel yeast gene. It's one of those genes that's like the LAC-Z I told you about when we did the lac operon. It's so useful that you see it in many molecular biology applications. So we'll talk a little bit about it as an example of a transcriptional activator in eukaryotic cells. Okay. So as I said, we'll just go through the first few slides that we did last time very quickly just to kind of bring us back to where we're going. So we're looking at eukaryotic gene regulation. What we really want to do is look at what are the similarities where you kind of see an evolutionary link, but also where are they very different. Here's a key one again. Eukaryotic genes do not have these operons. This idea that you take genes that are related that need to be expressed for some common function, such as synthesis of an amino acid, cluster them together, put them under control of one switch, turn it on and off with the lac operon being the model. You don't see this in eukaryotic cells. Also, eukaryotic mRNAs are monocistronic. That simply means that each mRNA codes for a single polypeptide. In bacteria and prokaryotic systems, you have this polycistronic. So, for instance, in the lac operon, there's 3 structural genes, LAC-A, LAC-Z, and LAC-Y. There's one messenger RNA, but it codes for all 3 polypeptides. So this is polycistronic, being more than one cistron or more than one gene. The other is that we have to deal with chromatin in eukaryotic cells. So that we have to displace the histones, the nucleosomes. And, as always, eukaryotic cells need many more proteins to do a similar job. So If you think about sigma factor as a transcriptional activator, it really isn't. That's basically one. E. coli codes for 3, 4, maybe 5 transcriptional activators. In eukaryotic cells, there's probably, it starts numbering into the hundreds. It's not even worked out yet. Some people think maybe, upwards of 50% of all of our genes may be transcriptional activators. Okay. So again, just this idea that in this chromatin is in this very, you have this chromatin is this in a diffuse but still highly structured, highly condensed state in the nucleus during interphase. In order for the RNA polymerase complex, this basal transcription apparatus, to have access to the DNA, you've got to rearrange that chromatin. You have to move it around a little bit. This is what we talked about last time. And we can probe that. That is, we can look at it experimentally by looking at changes in the sensitivity of those DNA sequences to an enzyme such as DNase, which will, the rationale being, the DNA that's most accessible to the enzyme will be the first DNA to be cleaved. And when you do this, what you find is actively genes that are going to be transcribed, that are going to be actively expressed, like the tyrosine hydroxylase in the dopaminergic neurons, you'll find that they are now in a conformation that is hypersensitive to the DNase. Now, if we did that same question but said, what if we took the chromatin from the brain? Would you have DNase hypersensitive sites around the gene? No, because there, the gene's not expressed in the brain, and therefore the chromatin stays in this heterochromatic, inactive, non-transcriptionally accessible, compact state, which is not going to really be accessible to the DNase. So again, the enzymes are here. They're just a tool to help us probe and understand the structure. The other is this modification of the histones. The idea that the histones have basic amino acids, lysine and arginine, so they're positively charged at neutral pH, at physiological pH. That is going to do what? Interacts with and neutralizes the negative charge on the DNA. So that holds the histones tighter to the DNA. So one of the things we have to do here is release the DNA from the surface of the nucleosomes to again make it accessible. And one way to facilitate that is to knock out that positive charge. So that's what acetylation is doing. It's just simply, it's still a polar amino acid. It's still very hydrophilic, yet it loses its positive charge. Can't interact with the DNA as tightly, so now the DNA is not as tightly associated with the surface of the nucleosomes. So these events are all happening to help get the DNA in a conformation so it can be transcribed. In a regulatory sense, we also see DNA methylation. So this is probably the most common chemical modification to DNA. And in eukaryotic cells, DNA that is methylated is almost invariantly methylated on C residues. So it's at position 5, so you have 5-methyl-C. This is on the base structure, not the sugar. Okay. And what very commonly find is when that C residue is methylated, it's followed by a G. So that gave rise to this idea of CPG islands. This just means short stretches of repeating CG sequences where the C's are methylated, and that P just indicates the phosphodiester bond between the two bases. All right. So what you find is inactive genes are heavily methylated. Active genes are hypomethylated or have very low levels of methylation. So we go back to our tyrosine hydroxylase gene. That gene in the dopaminergic neurons would be methylated or not methylated, relatively speaking. It'd have fairly low levels. Let's say compared to the same gene sequence in the brain you would see higher levels of methylation because the gene is inactive. So you really have three things here now that are setting the stage for being able to transcribe the gene. You have a looser conformation of the chromatin. I think I left out the slide, but if you go back and look at the notes from last time, you literally have in many cases, the nucleosomes are repositioned by these chromatin remodeling proteins. The DNA is more loosely associated with the histones and the methylation all come together to facilitate the ability to transcribe that gene. All of this has to happen before we can actually get transcription of the gene. Yes?
If something's part of the sequence that in other cells would be transcribed, wouldn't it still be in an inactive place? It would still be chromatin and not heterochromatin? Because I thought you mentioned in the stomach heterochromatin.
Well, okay, if I'm following what the question is, what you're really saying is in a microenvironment, the same DNA sequences with the same position in the chromatin are in a different conformation in the cells where the gene is going to be expressed versus cells where it's not going to be expressed.
It would still be, it wouldn't be called heterochromatin.
Oh, you can't really tell that until you actually have the chromosomes condensed at metaphase. So this is still interphase, and so looking under the microscope, you can't really say heterochromatin versus euchromatin. Any other questions? Okay. All right, so this is where we left off. This is just one slide, just a couple of comments on epigenetics because there's a whole chapter on this, chapter 21. This is kind of rewriting, and I think in many ways, what we think about genetics. Anybody familiar with Lamarck, his ideas? They still teach that they teach you that in biology, and then they tell you it's all nonsense and it's all random mutations. Okay, well, it turns out Lamarck may have been right, but for the wrong reasons. There's now, this has come out in the last several years, many examples of so-called epigenetic events. All this really means is some sort of chemical process, biochemical process, whatever you want to think about it, that doesn't change the DNA sequence, but it changes the inheritable trait. And methylation is at the root of at least the parts that we understand. I think we're just beginning to understand how this works. So one of the first examples that was found was this behavior in behavioral biologists working with rats and such. Noticed that mothers that spent more time grooming the pups and licking them and such, as adults, those animals were less fearful and coped with stress a little bit better. When we get to the chapter on epigenetics, the initial story that is at the beginning of that chapter is really quite interesting, and it presents evidence that, I don't want to depress you, if your grandfather grew up in times when things weren't stressful, there's plenty of food. Let's say he was in like my grandfather grew up in a farm, and so if it was a time when the weather was cooperative and there was plenty of food, the grandchildren actually have a higher incidence of developing diabetes, some other health-related issues, and tend to have a shorter lifespan. If your grandfather had it rough and food was hard to come by and he grew up under some stressful conditions, the grandchildren live a little bit longer. So the question is, how could the behavior of your grandfather actually affect you 2 generations later? So that's some of what we'll be talking about when we get to that chapter. Okay, so here's really the new material starting up where we left off from before about transcriptional control. First, we'll kind of review some of the information from chapter 13 because you got to go back. And you want to think about this, it's a structure dictates function. We got to go back and look at the structure of this transcriptional apparatus and then talk about how it's regulated. Okay. Now, before we get to that diagram that you've seen before, the key player here is going to be these transcriptional activator proteins. So they bind to a specific DNA sequence. We've talked about this before. Now, to look at them a little more carefully, in many cases, they're a bifunctional molecule. One end of the molecule interacts with the DNA. The other end of the molecule facilitates protein-protein interactions. Now, so what does it interact with? If it interacts directly with that basal transcription apparatus, what does it interact with? You're going to wait for me to tell you, right? Remember there was that mediator protein? So I'll show you that on the diagram. Or you can have a coactivator. Think about it this way. The coactivator is just a link. So instead of that you've got this transcriptional activator protein bound to the DNA. Instead of it interacting with the mediator directly, there's another molecule between there, another protein. Now, will that one have a DNA? Will that co-activator have a DNA binding domain? No, it doesn't bind to the DNA. It interacts with the transcriptional activator on one end and the mediator on the other. So it forms a bridge that still connects the transcriptional activator to the transcriptional apparatus. Okay. Also, you kind of remember there's two components to that promoters. So we have the core promoter, which is really where that basal transcription apparatus binds. Then the regulatory promoter are those upstream sequences which bind these transcriptional activators. Okay? And so these binding sites for the transcriptional activator are located upstream of the core promoter unless they're not. Okay? Because then we have these enhancers. So enhancers are essentially doing the same thing. They're doing the same job. The transcriptional activators are binding to them. However, they have more flexibility in their positioning. So sometimes it was very confusing and took a long time to work out the story, because sometimes the enhancers would be located upstream of the promoter where you would expect them to be, although it might be thousands of bases. Other examples were showing up downstream beyond the end of the transcript. And then more recently, some examples are showing up in the introns of the genes. To make it even more complicated, there's at least a couple of examples in Drosophila where there's a gene located within the intron of another gene. But we won't go there. Okay. So here's that picture we've seen before. And you should be very familiar with this because you studied all of this and you're ready for all of this last Monday, right? But now that you've cleared the slate so that you can start the new, now I'm confusing you by bringing it back again. Okay, so here's what we had. Here's the transcriptional start site. Here's that TATA box. So we have the TATA box binding protein. Basically is the peg that positions the whole transcriptional apparatus in the correct place. The core promoter is where this basal transcription apparatus is going to associate. And then upstream are these regulatory promoters. So this is the basic transcriptional apparatus that you're going to find on any one of these genes, but unless it's activated by some interaction with this mediator, it's really just going to sit there. It's just parked and ready to go. It needs to be activated. Here's one of those transcriptional activator proteins. It binds to its DNA sequence that's up here on this regulatory promoter. And in this case, there's a co-activator, so that you have a protein-to-protein interaction. Or you can have another transcriptional activator protein that binds to an enhancer. So what's really the difference between this sequence and this sequence in functionality? Okay. These that are part of this regulatory promoter tend to have to be very position-specific and also orientation-specific. You had a mutation that inverted it, it wouldn't be functional. The enhancers, since the DNA is flexible, they can be upstream, downstream, anywhere, and they seem to function whether you flip them over or not. They're pretty much orientation independent. But they're both really doing the same job. So now, this is what has to assemble to initiate transcription of this particular gene. Without both of these transcriptional activator proteins, if you only had one of them here, do you think you'd get transcription? No. In this case, you need two, but sometimes it'll work with just one. I'll show you why. So it's a way to be able to regulate the expression of the gene under different metabolic conditions. Okay. So as probably the simplest example of a transcriptional activator in a eukaryotic cell is this Gal4 protein from yeast. So it's a transcriptional activator, and it regulates transcription of genes involved in galactose utilization, just another sugar. As I said, we've used it. It's used very widely in molecular genetics. It's just one of those very useful genetic systems that we know a lot about. Okay, so it's a transcriptional activator. So that protein is going to bind to a specific DNA sequence. In yeast, these sequences that are in the regulatory promoter are called UASs. And that's just for an upstream activator sequence. Just means sequence is a DNA sequence that a transcriptional activator is going to bind to, and it's upstream of the structural gene. So this binds to UASG, which just simply means it gets its name from upstream activator sequence for Gal4. It's a transcriptional activator, so the binding of the Gal4 activates transcription. So we wanted, it's not a good comparison, but if we wanted to compare it to those 4 different ways of regulating gene expression in prokaryotes, would this be positive or negative? It would be positive because the binding of the protein activates transcription. And would this be inducible or repressible? It would be inducible. Does that make sense? This is very much like the lactose. The yeast don't want to express these genes unless there's galactose present. So there's no reason to make them all the time. You just want to express it when the galactose is present. So the Gal4 binds to this upstream activating sequence and turns on gene expression. But it's never quite that simple. Okay. There's another protein called Gal80. Gal80 regulates the expression of Gal4. So if you don't have galactose present, and I'll show you in a figure in a minute, the Gal4 can bind to its DNA sequence, but Gal80 binds to the Gal4 protein and blocks it from interacting with the mediator. So that's the situation you have when there's no galactose present. When you add galactose, then the galactose binds to the Gal80, involving another protein called Gal3. We'll get to that in a minute. But what happens now is it's very similar to what you saw in bacteria. The galactose is binding to the Gal80 and in the end ends up changing its shape to no longer bind to Gal4. Gal4 interacts with the mediator and turns on gene expression. So here's the way it works. Take the same system we had before. Here is the Gal4 protein functioning in. Why are there 2 of them here? 2 proteins. Because the artist just got carried away. Many of these transcriptional activators function as dimers. They function as pairs. So you really have 2 copies of Gal4 is bound, but Gal80 is essentially coating it and preventing it from interacting with the mediator. So now in the absence of galactose, the whole basal transcription apparatus is there, it's ready to go, but you have no transcription. Now when you add galactose, which is the effector molecule, the inducer, it actually binds to a protein called Gal3. Eukaryotes just have to do this in a more complicated way. So now you have galactose bound to Gal3, bound to Gal80, and the binding of the Gal3 with the galactose causes a conformational change in Gal80. It no longer binds to Gal4, and now Gal4 is free to interact with the mediator and turn on transcription. Okay? And that's probably about the simplest system available in eukaryotic systems. This is the lac operon of eukaryotes. Okay? In many ways, very fundamentally similar, but has a few more players and works in some other there's some other differences. But basically, you can see sort of that evolutionary connection between prokaryotes and eukaryotes. I'm sorry. All right. Let's see if this made sense to you. Here you go. Try that. I'll go to 1:15 on this one. Yeah, this is probably a very good example of a likely exam question. Okay. All right. So what's the answer? Never know if that's good or bad on this one. All right. So does it make sense? So this is a temperature-sensitive mutation. So you have a mutation in GAL80 so that when you grow it at the 30 degrees, the GAL80 is non-functional. It can't bind to GAL4. Well, if you go back here, that's the same as having done this. If a GAL80 can't bind to GAL4, then the GAL4 is free to interact with the mediator, and you would have constitutive expression of those genes, even whether there was lactose in the media or not. Okay. All right. Any questions? All right. So we've talked about these enhancers, and one of the potential problems is maybe the enhancers are positioned in a way in which it might actually affect expression of two different genes, or genes that that the cell doesn't want it activated at that time. And so this idea of insulators are really just boundaries. They're going to be DNA sequences where proteins are going to bind and basically just work as a molecular blockage so that the enhancers binding their transcriptional activator only work with the gene that's needed. So that's what these boundary, again, these insulators, when you see boundary elements, since you're seeing elements, it's telling you it's a DNA sequence. Okay, and it's probably easier to see it here. So what you have is an enhancer, which could potentially, like enhancer 1's over here and enhancer 2's over here, could potentially interact. Each one could interact with each of these two genes. The idea of these insulator binding proteins, so the insulator is a DNA sequence, it's an element, the protein sequence binds to it, is to help ensure that the enhancer is only turning on expression of the appropriate gene. Okay, any questions? All right. So here's that question of coordinated gene regulation. So we saw this operon model in bacteria where you have all the genes are already clustered together and they're under control of one promoter. You don't have that in eukaryotes. But sometimes you have certain situations where two or more genes need to be expressed at the same time in a coordinated way. And one of these is the so-called heat shock response. So if you thermally stress cells, what you find is there's a very rapid increase in the expression of about 20 different proteins, the so-called heat shock genes. So the idea is, how do you coordinately turn on expression of all 20 of these genes since they're located throughout the genome? So you need to have some common element. You got to have some common signal that can be turn on expression of all 20 genes. And that's exactly what you have. In this case, we'll call it a response element, but it's the same thing. It's a DNA sequence that's now located in the regulatory promoter of each of these 20 heat shock genes. And now the same activator protein is going to bind to that same element and turn on expression of all 20 genes, even though they're scattered throughout the genome. Okay. So that's what you see here in cartoon form. So here's that same protein we've been seeing. This happens to be a metallothionein gene. So metallothionein is an important gene. One of its major tasks is to help protect your cells from toxic metals. So hopefully not, but if you get exposed to cadmium, mercury, some of these toxic metals, turns on expression of the metallothionein gene, and that protein helps get rid of the toxins. So what you see is, here's that this portion is that upstream regulatory sequence. And you have all these different DNA binding sites, and each one can bind one of the activator proteins. So what it does is it now gives you an opportunity to either precisely control expression of the gene by needing two of these transcriptional activators to be present at the same time, or you can turn on gene expression under different physiological conditions or environmental conditions within the cell. Okay. It's not important that you memorize the names of these elements. I'm not going to ask you that. It's more important you understand how the system works. There's way too many of these to, We're going to try to memorize these is like just the tip of the iceberg. So, besides, you have this thing called Google. You can find them all at your fingertips anyway. But you got to know how it works. Okay. So if we kind of look at this, these things I've been talking about, we can look at some just common features now that we see in transcriptional control in genes. Okay. One is this idea that a single gene can now be activated by several different response elements. We talked a little bit about this before. The idea that, okay, humans have about a little over 200 or so different cell types. Well, so you could say I need to change the expression of a particular gene in 200 different ways depending on the cell type. Well, if we have a DNA element and a protein binding to it, we have two states. Either the protein binds or the protein's not there. But all we need is 8 elements, and we have 2 to the 8th or 256 different regulatory states. So by having just as few as 8 elements, you could have enough different ways to regulate gene expression in each of the cell types within the body. Okay? Or again, by having, so it gives you this much greater control over the gene expression as opposed to the operon, which is pretty much an on or off system. Okay. Also, having these multiple response elements means now I can turn on gene expression under different conditions. So the heat shock genes were first found named because they were found because of thermal stress. But they get turned on by a number of other environmental stresses, anoxia and such. So there's hypoxia elements. There's other DNA sequences that bind other transcription factors that turn on the same genes but under different environmental conditions. Okay. All right. Any questions about that? Everybody just goes, yeah, got it. Okay. Another thing we've talked about before is this differential splicing that all came out. We talked about in a very general sense. What I want to show you now is take that example and a way in which it's actually used to regulate gene expression. This is going to come from Drosophila. It's one we've talked about a little bit before. It really sets the stage for what people call sexual dimorphism, or basically what are the genetic triggers that tell this embryo it's either going to develop as a male or as a female. And so it's going to start with our old friend SXL, the sex-lethal protein. The key here is the sex-lethal protein is only made in female cells. I'll show you why that's important in a minute. Sex-lethal is a protein that binds to an RNA sequence. So what sex-lethal is going to do is bind to the pre-mRNA for the transformer gene, and it's going to affect how that pre-mRNA gets spliced, whether it gets spliced in a female-specific pattern or a male-specific pattern. Okay? So in the absence of SXL, so that would be in male cells, the splicing of the mRNA occurs in different places. So in females, you get splicing that gives you a functional TRA protein, transformer protein, and in males, you get a non-functional protein. Then that TRA protein can move on to the next step, which is probably easiest to see in this figure. So if it's an XX genotype, And so the trigger for whether or not these cells express SXL goes all the way back to that X-to-A ratio. So if the X-to-A ratio is 1, as we talked about before, which would be an XX genotype if you have the normal diploid number of chromosomes. And we won't go into the mechanism that triggers that. It comes as chicken and egg situation. We keep going back further in time. So in female cells, or at least cells that have an XX genotype, or an XXY, remember, because that doesn't affect the X-to-A ratio, they make the SXL protein. That's going to cause the TRA pre-mRNA to be spliced. I'll show you where this is, what I mean by downstream. But it gives you a functional, the result is a functional TRA protein. That protein interacts with the TRA2 protein to affect expression and splicing of a pre-mRNA called double sex. Okay. And if double sex is doing its job correctly, then it turns on all the genes needed for female development. In an XY genotype, there is no SXL protein. So the splicing of the pre-mRNA occurs in a different site that ends up leading to a non-functional TRA protein. Without the functional TRA protein, you don't have functional double sex DSX protein, and that leads to the male developmental pathway. So all of it is being controlled by the presence or the absence of the SXL protein and where that protein binds. So this is the TRA. So this is the pre-mRNA from the transformer gene. So initially when geneticists were looking at that, they just expected that in males they wouldn't express the transformer gene. But in fact they did. So they make a perfectly functional pre-mRNA. The difference is where it splices. There's two splice sites. This first arrow right here and this one. The key is in exon B, there's a sequence that leads to premature termination of the polypeptide. If this is part of the mRNA, you're going to get a non-functional protein. So now, let me see it. Okay. So if it splices here, then we get this mRNA. There's a nonsense codon in here that gives you premature termination of the polypeptide, and you get a non-functional protein. If it splices over here, it bypasses it splices from A, right? So it splices around it, and you get A, C, and D, so you're not including the B exon. You splice around it. Then you don't have that nonsense codon, and you make a functional protein. Okay, so what's SXL doing? This is the preferred site. So there's two sites here for splicing. Molecularly, this one is more attractive, we'll just call it that, to the splicing mechanism. So if it's available, the splicing is going to occur here. The SXL protein is binding over that site. Okay. So now it's going to splice here. Gives you the B. You get translation, and you get a non-functional protein. Without the SXL protein, with the SXL you get this pattern, you get it with females. Without it, you get male. So it's just simply being controlled by whether or not that first site is available. If it's not available, then splicing occurs at the second site. Stunned silence. Make sense? I know I say that a lot. It's a very kind of mechanical approach, yes.
How does it relate to the X-to-A?
Okay, the relationship is if the cells have an X-to-A ratio of 1, they express the SXL protein. So it'll be present. So you'll get this will happen in female cells. The SXL protein will cover up the RNA and block this splicing site. So in females, they have to splice at this site. Okay. And then that leads to this pattern.
Why does the ratio affect when splicing?
Oh, no, it just controls, it controls whether it makes the SXL protein or not. In males, you're missing the protein, and in the absence of protein, this first splicing site is the one that's preferred. Okay. Last little comments. So it's a fairly short chapter. RNA stability. We've mentioned this before. In bacteria, you're pretty much told you get transcription, you have translation of the mRNA, then the mRNA is degraded. And the mRNA is only around probably for a couple of minutes. And for most genes in bacteria, that's probably true. In eukaryotic cells, that can be true, or the RNA can be stable for hours, it can be stable for days. It depends on the tissue, depends on the genes being expressed. And the stability is controlled by more than simply whether or not nucleases in the cell are just going to degrade this mRNA. One of the unexpected findings of, well, probably about 15 years ago now, was the discovery of RNA silencing, or you may know this already as RNA interference. And that is, these are small little microRNAs that function, this is a form of gene regulation because they control whether or not the messenger RNA is present. So instead of degrading an mRNA just randomly by nucleases within the cell, this is a genetically programmed system to target that messenger RNA and degrade it. At first, it looked like it was just going to probably affect just a limited number of genes. Now it looks like probably a third of all the genes are affected by RNA silencing. It also became an extremely valuable tool for cell biologists working in tissue culture. And so this was one of those cases where its value was so obvious the Nobel Prize was awarded just a couple of years after the discovery. There's a new system we'll talk about later on called CRISPR that probably may fall into this same category. Okay, so here's how the fun starts. The idea is the cell is going to make a double-stranded RNA that is complementary to whatever messenger RNA is targeted for. So it's the mRNA for your favorite gene. If it needs to be selectively degraded by this mechanism, the cell makes a double-stranded RNA. How could it do that? Do cells express double-stranded RNA? So how do we get here? You could have inverted, sometimes it's inverted repeats. So it'll be a single RNA, but they're inverted repeats, and they'll snap together and form the double-stranded RNA. Sometimes it's expressed as two complementary sequences, and the RNAs anneal together in the cell. But in one way or another, there's a double-stranded RNA, and then it's cut up. I didn't make this name up. It's cut up by an enzyme called dicer, and it cuts it up into short, small pieces, and the pieces now are so short, they're only held together by the hydrogen bonding. It's not stable enough under physiological conditions, and it breaks up and you get these short, little, single-stranded interfering RNAs. These are the little RNAs now that are going to be able to, okay, so you got double, there we go. Got the little small interfering RNAs. They associate with a complex called the RISC complex. And together, this is the little small interfering RNA that's complementary to the mRNA sequence and associated with RISC. It binds to the mRNA, and then it leads to degradation of that RNA. So this is called gene silencing. It's not affecting transcription. It's affecting the stability of the messenger RNA, and it's a genetically controlled, programmed way to selectively degrade messenger RNAs. All right. And the other, to kind of just wrap it up, is even after we have all these levels of gene regulation. We got initiation, we said that initiation of transcription is the rate limiting step. Okay, but then you got the processing step. You got different ways of putting the exons together. Then you've got this RNAi interference that we just showed you about. Then there's translational controls. There's also regulation on how fast the gene gets transcribed. And then even after the protein is made or during the process, there's additional levels that can come into play. And that's what we mean here by translational and post-translational controls. Many polypeptides, as they're made and as they come off of the ribosomes, are not functional. They have to have additional biochemical events, modifications to them. Some of them have to have some amino acids clipped off. We said that all prokaryotic and eukaryotic proteins start with methionine. But if you purify that protein, in many cases, the methionine not there. It's already been clipped off as part of this post-translation or post-synthetic modifications. Or you may have glycosylations, phosphorylations, acetylations, different chemical modifications that happen to it at that point. So all of these events have to come together before you finally have a functional protein in the cell. And so now much of the research is focused on looking at more carefully how this regulation occurs and what those downstream target genes might be. And so most of the big story is, well, there's still a lot of proteins to be investigated, but it's also how they get expressed. So what we'll see later on is, for instance, the oncogenes, the cancer genes. These are normal genes in your genome, and they are expressed. They're called proto-oncogenes. Somebody won the Nobel Prize for figuring that out, a guy named Steve Bischoff. But if they're expressed in the wrong tissue or at the wrong time or in the wrong amount because they control cell proliferation and such, they are associated with a cancer process. We'll talk about that when we get to that chapter. Okay? So now that I got you mesmerized by that, there's the answer to that. So let's go back. Okay, so let me put in one slide. So see if you can figure this one out. You have, I'll give you about 15 seconds. Wonder where the hell that slide went. Oh well, nobody bats 1,000. Well, you guys read fast. Okay. So did I get it right? Sometimes, I don't know. Sometimes. You guys have caught ones that I got wrong before. All right. Post-transcript. Okay. Okay. Any questions? Okay. How many thought that was complicated? More complicated than neurobiology? Good, good. Wait till we get to the next chapter. No, I'm not. I'm trying. Am I trying to scare you? Yes. Don't worry about it. Okay. We'll figure it all out. All right. So that was the chapter on gene regulation. So we looked at, in terms of, we'll call it DNA metabolism. We've looked at DNA replication. We've looked at transcription, RNA processing, the translation process. The third area of DNA metabolism that we really need to look at, and as I said, one that's probably only been talked about a little bit in your previous classes, are the areas of genetic mutations and DNA repair. Because the first overarching idea is DNA mutations go on all the time. It is not a rare event. Right now, as you're tolerating me for the next half hour or so, we're each going to lose a few thousand purines in our DNA. Fortunately, or hopefully, you have the repair systems are in place that are constantly repairing the DNA. So there's actually a fairly high level of DNA damage that occurs just as a consequence of the biochemistry going on and the fact that we're all these elevated incubators running all these reactions at 37 degrees, and some of those reactions start running amok, and we have damage to DNA. But fortunately, you have these very, very good DNA repair systems. If you don't, you have a problem. There are a number of cancers that are very clearly associated with defects in specific DNA repair systems. We'll talk about that a little bit later as we go through this. So there's really going to be 3 parts to this chapter. And it's a long chapter. We're not going to cover it all today. It's probably one of the longest chapters that we'll cover. The first is just what do we mean by mutations, and to look at what types of mutations can occur, and then the second part is how those mutations can happen, and the third part is how do we fix it. So today, for this first part, we'll really be looking at what do we really mean by mutations and some of the ways in which this happens and the type of events that happen. Okay, so at a very broad basis, by mutation, we're just going to mean any inherited change in genetic information. Let's be specific here. These are going to be inherited changes in the nucleotide sequence. The statement has to be refined a little bit within the context of epigenetics. So we're talking strictly about changes in DNA sequences. Do we want any DNA mutations to occur? Would it be better if there were no DNA mutations? No, it's an evolutionary mechanism. It allows some changes in alleles and sort of testing out to the next generation. The problem is too many mutations too quickly is deadly. So we can tolerate a small level of mutagenesis, but the amount that goes on just normally, even in the absence of anything that we've done to the environment, would still lead to a too high a rate of mutation. And if that were true, you wouldn't actually have to be sitting here listening to me, because we wouldn't be here anyway. All right. The other is the real reason for genetic mutations is so that people like myself can have a job. So going all the way back to Mendel 150 or so years ago, genetic mutations have been a very powerful tool for the biologists, the molecular biologists, the biochemists, and such. So we've been giving you all these examples really in almost a historical sense of what's been going on for the last 150 years. And without genetics, this all would have been much more difficult to do. So I don't think it's surprising that you see, as the technology changes, the most useful model organisms, like Drosophila, are the ones that have good molecular biology, but also have very good genetics behind them. Okay. So first, let's make a distinction between somatic mutations and germline mutations. So the somatic mutations, you know what somatic cells, these are the diploid cells that are not part of the germline, that are not responsible for sperm and egg production, so that any changes in somatic cells will affect that individual potentially, but are not transmitted to the next generation. And what we see is a lot of the cancer mutations are these type of somatic cell mutations that are occurring because of some random event in the environment or some other process, but they're happening in cells that are not involved in the germline, therefore they're not transmissible or inheritable to the next generation. When this happens, if we have a genetic mutation, later on I'll tell you about a type of tumor that appears, if you did a pedigree analysis, it appears that it's a dominant mutation. That is, if you have one copy of this mutant allele, your chances of developing the tumor is literally 100%. But in fact, it's a recessive, and it all comes down to random mutations that occur within the cells. But I'll give you that. That's my little teaser, like the movie trailer sort of thing. It's what brings you back next time. All right. If you have a somatic mutation, you can have a clonal population. So just like when we talked about X inactivation to form the bar bodies, once that was decided, whatever that mechanism was, all subsequent cells would inactivate the same X. So when you have a somatic mutation, all of the cells that are derived from that cell through the process of mitosis will now have that same mutation. So we call this a clone of the cells or a clonal population. Okay. Makes common sense that the earlier in development that mutation occurs, the larger that clonal population. In general, just very ballparkish, somatic mutations occur about 1 per million cell, about every million cells, every cell cycle. Okay, that doesn't sound like very much, but it also means that we each carry about 100 million somatic mutations. That's pretty much generally. Right now, it's pretty much estimated that they've done enough meta-analysis of the human genome that, on average, we probably carry about 1 or 2% of our genes have recessive mutations in them anyway. So for those of you who think you're perfect, you're not. You carry a lot of mutations here. All right. Most of these have no phenotype. You don't know they're there. You have no observable effect. But, as I said, in certain unfortunate events in one of these cancers, when that happens, depending on where that mutation happens, then this can be a trigger for oncogenesis. Okay. Germline mutations are going to be a little different because these are now going to be inheritable mutations. I just put this in here to clarify. We've talked about chromosomal mutations, inversions, deletions, translocations, and such. Those are very, very large scale. Here we're talking about single cell or probably a single gene level type mutations. So just don't confuse what we're talking about here in terms of gene mutations to these large scale chromosomal mutations that we talked about before. All right. So with that little bit of an introduction, let's try this question. Okay. Oh, 1 minute should be good. Okay. So what's your favorite answer? All right. That was just to give you a brief. Okay. All right. So now we can start talking about this next section is on the types of mutations. The first group are the ones you're probably a little more familiar with, involving single nucleotides. So the kind of the jargon term we use is a base substitution. So base substitution just simply means that a single nucleotide in the DNA sequence has been changed to another nucleotide. We can have transition and transversion mutations. Transition is just a like for like. A purine gets changed to another purine, or pyrimidine gets changed to another pyrimidine. So transitions, like to like changes. Transversion is just the opposite. A purine gets changed to a pyrimidine, or a pyrimidine gets changed to a purine. Okay, later on we'll see which of these types are more frequent, but for now we'll just focus on what we mean by these two types of mutations. So for the transition mutations, that gives us 4 possibilities. We can have A to G and G to A transition mutations, or T to C and C to T. And for the transversions, then there's 8 possible mutations. Okay, that's all we mean by that. All right. Some of these phenotypic effects, these are some you're going to be familiar with already, a few of these. So we have the missense mutations. That just simply means we're going to be changing. What does that mean? You guys know? Okay. We're just changing one codon. We're changing a codon so that it codes changing from one amino acid to another. Will this always be detrimental? No, that's what we talked about before. If the biochemical property of the amino acids are similar, then you're less likely to have an effect than if they are quite different. Okay? Nonsense mutations really mean you're changing a codon that codes for an amino acid to one of the 3 stop codons. So nonsense meaning it no sense it doesn't code for an amino acid, but these are stop codons. Those do nonsense mutations probably have a significant phenotypic effect? Yes. Maybe. Depends on where it happens. Would it be more, if it occurs closer to the 5' end of the mRNA, would that be more or less likely to have a strong effect than if it were close to the 3' end? Right. So one is it really has to be in the open. Well, most of the time it would be in the open reading. It would have, in this case it would be. I was saying that. Okay. So it's got to be in the open reading frame. The closer it is to the end terminus, the 5' end, because it's going to be a premature termination, the more likely you've probably knocked out the functionality of the protein. You could be very close to the C terminus end and only delete a few amino acids and maybe have no effect on the protein. But in general, what's going to be worse, a nonsense mutation or a missense? Yeah, I'll take my chances with the missense mutation. All right. A silent mutation is just a base substitution that changes the codon, but it doesn't change the amino acid. So remember we said the genetic code is degenerate, and that except for tryptophan and methionine, all the amino acids have more than one codon. So a silent mutation just changes one codon to another codon that still codes for the same amino acid. So would that affect the functionality of the protein? Was it really a mutation? Was that one of those if a tree falls in the woods sort of things? Okay. So did the mutation happen? Yeah. It's just the phenotypic outcome, we couldn't find it. All right. Could you find that mutation? What would you have to do? You sequence the DNA. Would sequencing the protein do you any good? No, you'd have to look at the DNA sequence. Okay. A neutral mutation is the other type of changes that we talked about, and that is to change amino acids from one to another amino acid that has a similar biochemical property, and therefore it'll probably have minimal impact. Yes?
A silent mutation wouldn't affect like somatic cells, because it's like the same thing, it's not changing proteins, right?
Right.
But if it was meiosis and maybe it got mixed around in a different way with something else, would it then have an effect like in future generations? Like maybe depending on the way it's replicated?
Yeah, I'll say, I don't like to rule anything out, but I I wouldn't think so. But see, now I'm going to be get so easily distracted now. I'll be thinking about it. All right. Okay, so let's try this question. So everybody's going, I used to know those. Wait a minute, was that 2 exams ago? That was... No. Wipe the slate clean, move on. All right, I'll go to 1 minute on this one. Okay. What's your best guess? Okay. So this is a transition mutation, so that's like to like. Okay. So the only one up there is G to A. Okay. surprisingly, it turns out is insertions and deletions may actually be more common than these base substitutions that we were talking about. So what we mean by insertions and deletions are the actual addition or loss of nucleotide sequences. So this is how frame shift mutations occur if these deletions occur within the open reading frame. Okay, so maybe a good question could be, there's been a deletion mutation. You have a deletion, you have a one-base deletion in the 5' upstream untranslated region. Let's say 8 nucleotides from the start site. I'm making this up as I go, so stay with me. What I'm trying to do is make a point. If you had a deletion at that point in the 5' UTR, could that change the protein that comes out of that process after transcript? Then you have transcription, translation. Would that be would that mutate? Would you expect to see a changed polypeptide? Well, what would it do to where transcription started? It would change. It would change. Actually, deleting it makes it. Yeah, it starts a little further downstream. So what I'm trying to do is just keep in mind that so many of the types of mutations that they've talked about in previous classes, they're really talking about the open reading frame, the sequence that specifies the protein the amino acid sequences. But we could have mutations in promoters, we can have mutations in these upstream untranslated regions and such that can affect how that gene is ultimately expressed. Okay, so we can insert or delete a base. Everybody understands how that leads to frame shift mutations, right? Okay. But what if the insertion or deletion is a multiple of 3? The codons are 3 bases. So you insert 3, let's say you delete 3 bases from the open reading frame. What's the consequence? You'd miss 1 amino acid, but what about the rest of the amino acids? They would stay in frame. So if you're deleting or inserting in multiples of 3, there's a reasonable chance you may have little detrimental effect on the polypeptide. You may gain a couple of amino acids or lose a couple, whereas if you have an insertion of 1 or 2 or a loss of 1 or 2, it throws off the whole reading frame and you get a completely different polypeptide at that point. Okay. So that's what these so-called in-frame insertions and deletions. Okay. So that's what it's trying to show you here. So here's the original DNA sequence. Here's the typical kind of base substitution that you're familiar with. We changed 1 base, and what happens is we change a single codon. Here, this is just showing the consequences of inserting or deleting a base and how now you change the entire amino acid sequence. Okay. One you're probably less familiar with, but there's more and more examples of this being found recently, so-called expanding nucleotide repeats. Now the full story isn't known here. And there were up until just a few years ago, there were only a couple of cases, but as it shows now, there's at least 30 human diseases that are associated with changes in the numbers of repeated sequences within a certain gene. So here's an example. There's a syndrome called the Fragile X syndrome. This is the most common form of inheritable mental retardation, mental incapacitation, diminished capacity, whatever. I don't know what today's politically correct term is. Okay. It got its name, the Fragile X syndrome doesn't mean that if you look at the karyotype of these individuals that they have broken X chromosomes, but rather it's a somewhat artificial situation. Cells from individuals with Fragile X syndrome, when you grow the cells in culture, in plastic dishes, and you starve them by leaving out the purine and the pyrimidine that it needs to replicate for the DNA, then the chromosomes break, the X chromosome breaks in a very characteristic place. It breaks in a gene called FMR1, And what you find is this gene has a triplet repeat, a CGG sequence that's repeated no more than 60 times. That's the normal situation. Individuals who have the clinical syndrome, when you look at their DNA sequences, the number of copies of that triplet sequence has now expanded out into hundreds, if not thousands, of copies. Okay. And that the more copies you have, the more severe the syndrome. There's a general correlation there. Huntington's disease has something very similar to it. It actually affects the protein that's being made and makes the protein more toxic. It showed there's more and more examples of this change in this expansion of these expansions of these repeated sequences. It seems to be happening through meiosis. So what you get is an unconventional type of Mendelian inheritance. It's a little complicated. We won't go into it. How that mechanism happens, we're still at the point of trying to figure out that mechanism. But it does seem to be related to a significant number of human health conditions. Okay, let's try this question. It looks like that just needs a minute too. All right. So in the answer, okay. All right. So looking at a little more severe mutations. So this one I think is I don't like actually, this is a common terminology, but I think it can be a little bit misleading. So there's so-called loss of function mutations, which really just means that there's some functionality of the proteins that has been lost. All right. What happens with many mutations, especially in genes that encode, let's say, enzyme activity and such, is they're what we call hypomorphic. They still retain some of their enzymatic activity, but it's reduced dramatically. Maybe it has 10% of the normal enzyme activity. So what I like to do, at least, even though it's loss of function may say a partial, I like to use loss of function. We're talking about the equivalent of also a null mutation. These would be mutations in which, by any way we measure it, there is no functionality. There's either no protein being made, or if there is a protein being made, there is no functionality to it. It's a complete loss. So, for instance, in my lab, the two genes that we look at, we made large deletion mutations that took out the promoter sequences and a big chunk of the transcribed units, the transcription unit, including part of the open reading frame, so we would be very sure that these were null mutations, that these would be complete loss of mutation, loss of functionality. In fact, there's no mRNA made from these genes or anything else. So these are complete loss of function or null mutations. Okay. These type of mutations are usually going to be recessive. All right. They're losing some functionality, but as long as you have a wild-type copy, one wild-type copy, you can still make the functional protein. All right. What's the situation when that wouldn't hold? Talk to the little fly with the funny wings, the notch. So, except for in cases of the haploinsufficient. All right. So you do have cases where one copy of the wild type gene is not enough. But in most cases, so the vast majority of mutations are in fact going to be recessive. But not all of them. Some are gain of function mutations. These are going to be mutations which actually give a new functionality to the protein. Maybe the protein might have some toxic properties to it now, to the cell. Maybe it's turning on some gene expression that shouldn't be happening. Maybe it's a transcriptional activator that now activates the wrong gene. It's gained some type of functionality. Do you understand why those would be dominant mutations? You have a new functionality, you're only going to need one copy of that mutation to be able to see this phenotype. So loss of function mutations are invariably recessive. Gain of function mutations are going to be dominant. And then there's conditional mutants. So these are ones that you've seen before. There's different kinds of conditional mutants. The one you've seen before, the most common one, are temperature-sensitive mutants. And so these just simply mean these are mutations where under certain environmental conditions the protein is actually functional, and on other conditions it's not functional. So temperature-sensitive mean that if you grow it at the permissive temperature, then the protein has normal function. Will that usually be higher or lower than the normal physiological temperature for the organism? Most of the time lower. It's a thermodynamic problem. It's a mutation that makes the protein a little less stable. It starts to denature, but if you can lower the temperature a little bit, it'll still retain its conformation and have some activity. So most of the time, the permissive temperature is lower than the normal temperature. Okay? And the non-permissive temperature is when you see the mutant phenotype, which in most cases would be the normal temperature that you'd use for that particular organism. All right. So, for instance, an important area where you had to have temperature-sensitive mutations are dealing with haploid organisms. You're studying E. coli. You want to study DNA gyrase, and can you study a mutation of DNA gyrase in E. coli that is a null mutation? Well, let me write that question down. Is DNA gyrase essential? Can cells replicate without it? So if it's a haploid organism, if you inactivate DNA gyrase, it's lethal. The cells will never grow. So if you're trying to study a gene that's essential and it's in a haploid organism, you have to have one of these conditional mutations. So you have conditions where you can allow the organism to thrive, replicate, whatever you need it to do, and conditions where you can study the mutation. Okay. And then the lethal mutation, yeah, that's the one. I think I told you guys, I did tell you about my softball team, right? Didn't I? Yeah, I thought so. Okay. They'll all walk around, oh, I wasn't here that day. It was an eye clicker question, if you remember. No, it wasn't. She's, what was it again? All right. If you want to know about it, ask me later. Okay. Lethal mutations, we've talked about those before. That just means, this goes to that question of whether they're lethal, whether they're recessive or dominant. So my softball team's name was the dominant lethals. Okay? Doesn't intimidate anyone who's not a geneticist because they don't even know what the hell you're talking about. Okay? These just mean, really, these are mutations that are going to prevent the organism from growing and developing, and they're going to act very early in embryogenesis. And they're just not going to survive past the very, very early stages in embryogenesis. So they are, by definition, recessive, and so they're just called recessive lethal mutations. You usually can find them. You'll realize these. If you're using something like Drosophila, and you're expecting 4 different genotypes and you only see 3, it's because the homozygous individual is not present and probably means the gene you're looking at is recessive lethal. All right. So I tried to prompt you for this question by something I said a little earlier. And somebody's going to not like the answer, but
Do you mean like homozygous at the clinical level?
do I mean what?
Homozygous at like the clinical level?
We'll talk about it.
Okay.
I'm trying to make a point here.
Okay.
All right, we'll go to 1 minute here. Okay. Oops, there you go. Gave you a chance. Hit the wrong button. All right. So just trying to make the point that I've made several times as we've gone through the courses, we use this recessive and dominant, homozygous and heterozygous. We're taking 150-year-old terms and trying to make them work with what we understand today. So whether you're looking at this mutation that causes a silent mutation, you know, maybe no one heard the tree fall in the forest, but the tree still fell. Okay? So it's whether it's this is your question, is whether it's homozygous or heterozygous goes to what level are we investigating. So if we look at the phenotypic level, we call it homozygous. If we look at the molecular level and we look at the DNA sequences, we'll see the DNA sequence is different, and then it would be a heterozygous situation. So it's just this idea of having to take some of this terminology and reshape it to fit what we understand today. Okay. All right. Oh, now we're getting to fun stuff. Okay. Forward and reverse mutations. What you're familiar with are forward mutations. You start with a wild type, some mutation happens, and now you have a new phenotype. You have a mutant phenotype. So this is traditional genetics, everything from the first white-eyed flies that Morgan found in his lab to mutations that were deliberately made with different mutagens and such. These are all these classic type of forward mutations. You can have a reverse mutation, which will now take that mutant type. You have a second mutation, and that mutant type now goes back to being wild type. So forward is we start wild type, we have some mutation, and we get a mutant phenotype. Then another event happens, and now we have wild type again. So now we have a reverse mutation. All right. So in this rather straightforward, non-busy slide, so here we have a wild type red eye color. So we have two genes here, wild type for A and B, and we get the wild type mutation. We have a forward mutation that mutates A, and you get the white eyes. We can have a reverse mutation, and this happens a lot with single base mutations. You can have a mutation that will now change that mutated base just by chance back to what it was originally. And that brings you right back to the wild type. So that's the reverse mutation. But because I like to make things complicated, we can also get a wild type eye color through a suppressor mutation. The suppressor mutation is going to be a second mutation, not simply reversing what happened the first time, but a second mutation, so that now we have a mutation in both A and B, and those two mutations together give us a wild type phenotype. Okay? And then I'm going to have to show you how there's two different ways you can get suppressor mutations. Okay. Okay. So, suppressor mutations mean we have a mutant phenotype, we're going to have a second mutation, and we're going to get the wild type phenotype back. The first one is an intragenic suppressor mutation. Okay? So we can probably use this also to kind of also show you the forward and reverse. So here's the wild type sequence, and you get this protein, and here's leucine. Then you have a mutation that changes, I think, what looks like a T to an A, and now it codes for phenylalanine. All right? What type of mutation? Is that a missense, silent, nonsense? That's a classic missense mutation. All right? Okay. Now, an intragenic suppress. Now, if we had a mutation that now we have a second mutation that mutates this A back to T. Okay? That would be a reverse mutation because you're restoring it back to the wild type sequence. Or we can have an intragenic, which means within the same gene, a second mutation that now changes one of the nucleotides. So now the sequence is GA. Is that a G or a C? I can't quite. GAA. But that codon also specifies leucine. So we've had two mutations in two separate events that brought us back to leucine. So the outcome here is the same. If this was just changing this base back to T, it would be a reverse mutation. This is two separate mutations that lead to a wild type phenotype. So this is a suppressor mutation. Okay. So it's a second mutation within the same gene that yields a normal phenotype. Okay. And in about the last 3 to 4 minutes, I'm going to show you the next one, which I think you're going to want me to go over again when we come back on Monday. All right. And that's intergenic suppression. This is going to be mutations in two different genes that bring you back to a wild type phenotype. All right. The difference being it's a mutation in another gene. All right. I don't know if I can figure this one out. So you have a mutation, just like we just did. You have a mutation in one gene that changes a codon and gives you an altered polypeptide, and you have a different phenotype. The suppressor mutation is going to be a mutation in another gene. In most cases, this is going to be a mutation in a tRNA gene. So let's say we go back to this one. And so we have a mutation that changed leucine to phenylalanine. All right? The intergenic suppressor would be a mutation in a gene that codes for a tRNA for phenylalanine, but it changes the anticodon, so now it recognizes. Wait a minute. That's right. I'm sorry. Erase that. It would be in the gene, one of the genes for the tRNAs for leucine. It gets a mutation in the anticodon that now allows it to recognize the codon for phenylalanine, but it's still charged with leucine. So that tRNA is going to insert leucine instead of phenylalanine. So in this case, two wrongs actually make a right. All right. So that's what this is trying to show, is that you have a mutation that changes the anticodon of a tRNA so that now it actually inserts the correct amino acid. Okay, that's not exactly, in this case, what it is, what happens when you, usually, those kind of, in a practical sense, those kind of mutations are hard to find these types of suppressors. What they usually are is suppressive for nonsense mutations. So those are the best examples. You have a mutation that causes a nonsense mutation. You get premature termination of the polypeptide, you get non-functional polypeptide, and you have some mutant phenotype. But then you find in another genetic background, you have a perfectly normal phenotype. And what it is, what you find is it's one of the tRNAs has an anticodon mutation, which now allows that amino acid bearing tRNA to bind to that nonsense codon and actually inserts an amino acid in there and prevents the premature termination. Okay?